Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Eugenia, and I'll be your host. It's a pleasure to have Solvius with us today, who will be presenting this webinar titled Biopharmaceuticals, Cell and Gene Therapy, Single-Use Technology, and Extractable Leachables. Is your product adulterated? Today's guest speakers are Christopher J. McGee, PhD, and Christopher A. Letandres, PhD. Team leaders in extractable leachable chemistry at Solvius, USA. Now I'll provide some housekeeping information before we start. First of all, I would personally like to welcome you to our webinar platform ON24. You will notice that this webinar is browser-based. So if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. During today's event, there will be plenty of time to ask our speakers questions. You can do that via the question widget on the left-hand side of your screen. Just type your question in this box and click Submit. We will allocate some time at the end of the session to answer questions or any thoughts that you may have. You can also review different types of resources connected to the topic of the event at the bottom of your screen. If you require any assistance, please use the yellow Help widget. In addition, you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. And now, please allow me to welcome Christopher Letandres. Over to you.
Thank you, Eugenia, and thank you everyone for your attendance and interest today. As we dive straight in, we have our agenda. Before we cover everything today, we'll cover definitions, regulations and guidelines, risks, container closure systems, single-use systems, best practices in the space, cell and gene therapy, and finally a summary. One of our first slides here, I think, demonstrates to us the complexity of not only the technical but the regulatory expectations in the EL space. We certainly won't cover each and every one of these today, but certainly leave you with an offer. If one of these is your question and you'd like for some further guidance or discussion on this, we're happy to oblige. You'll have our contact information on the final slide. Starting with some definitions here, extractables, we've got many groups in the space with slightly different definitions for extractables. And I think what you'll find here is some overlap in the concepts that are required to define our extractables. Certainly these extractables are chemical entities, both inorganic and organic in nature, but how we define them, how we generate them is different depending on the needs of the sponsor of record. And so concepts like how we extract, what solvents are used for extraction, how that informs potential leachables is a general topic that we'll discuss a little bit today, and certainly something that needs to be well understood by the experts that you work with in order to generate a compliant regulatory package. As we move on to other working groups here, the concepts of an accelerated or exaggerated set of conditions for the definition of your extractable study are critical to the design and really to understand the outcome. Each extractable study may have a different intended outcome, whether to inform potential leachables or even for the purposes of material characterization. So it is critical to know what questions you're trying to answer in order to design an appropriate extraction study. Moreover, these extractables are meant to inform potential leachables. And so the leachable definition here are really the questions of concern. These organic or inorganic chemical entities eventually can make it to your drug product and adulterate that drug product, leading to potential toxicity, safety, quality issues that need to be understood and quite frankly controlled from a quality standpoint. These leachables may originate from container closure system in some of the more classic leachable studies, but they may also originate, as we'll discuss further today, from process equipment. And those pearls, process equipment related leachables, are also part of this quality endeavor to ensure patient safety, but also have an opportunity to correct for any bad actors that do make it through that process stream into the finished drug product. For us as analytical chemists, we need to set those lines in the sand. The toxicologists set out a safety concern threshold, this SCT, which is a threshold by which a leachable would have a dose so low as to present negligible safety concerns for carcinogenic or non-carcinogenic endpoints. As we take that concept into our analytical chemistry on a chromatograph, we have to set up that line in the sand. That analytical evaluation threshold is calculated from the SCT, and it effectively says to us, where do we need to do further effort to identify and evaluate those extractables that have become leachables to better understand the impact to the patient. And you may have seen some of these types of diagrams before. Our extractable library should include all of the known additives that we can characterize in an extractable study. It would also contain any of the impurities that come in with those additives, whether that's also potentially monomers and polymers. And you can have some cross-reaction of materials and solvents during that study, and it's important to understand and segregate those. Ultimately, we're trying to answer the question of what then migrates into the finished drug product, and that would be our leachables library, those known extractables that have migrated. But there's also, because these are chemical entities, there's a potential that those are also modified. And so understanding some of the organic chemistry that can occur on those compounds as they do interact sometimes with drug product, whether that's API or excipients. I won't spend too much time here, but there's certainly a lot of very intelligent people in the field working in industry work groups to try to support prescriptive and suggestive guidance for efforts in the EL space. And ultimately, this all comes back to legal code. The regulatory authorities are enforcing legal code such as CFR Title 12, Part 211. You see terms here like in-process materials and components, and they have to be characterized and understood so that the drug products are not reactive, additive, or absorptive. And notice here the terms safety, identity, strength, quality, and purity of the drug product. While toxicology is a huge topic in EL, it's not the only topic. The identity, strength, quality, and purity must also be established and controlled for quality's sake. Title 21, Part 600 also calls out directly leachable contaminants that must be controlled so as to not render the product less suitable for its intended use. And those that follow the FDA guidance for process validation understand that this is a cyclic process, demonstrating that these materials are suitable for their intended use, and if they are not, or if there are any bad actors coming from that mitigating factors need to be applied, and that might be the replacement of a component or container closure component. Some of you may have seen iterations of this table over the years. This is a constantly evolving position by the FDA, comparing the degree of concern on the left for the presence of extractables and the likelihood of the packaging components and dosage form interacting with that container closure. And that would give us the presence of leachables. Ultimately, I think the takeaway from a slide like this is that the guidance and expectations are constantly evolving. The regulatory expectations can be uncertain, the technical complexity is certainly high, and there is a potential that as a sponsor of record, not engaging with experts in the field and addressing questions before they are asked can lead to CRL and potential loss of revenue. As we mentioned here, toxicology is one of the critical endpoints of EL testing, and there's two effects. Certainly the direct toxic effect of a leachable as exposed to a patient through the adulterated drug product is of major concern. And anyone working in an analytical chemistry in the space would be expected to engage with toxicology so as to help answer questions about the potential identification of a leachable compound. And certainly the toxicologist can take that information and generate some safety endpoints, whether that's a margin of safety or a PDE. The indirect effect of the leachable in the drug formulation could be as important. Impact on the potency of the drug product would certainly be a deleterious effect. At the bottom here, you see some ICH guidances, and I think it's critical for anyone in the space to understand that extractables and leachables are held to a separate standard. The working group that's continuing to work on ICH Q3E is trying to further call out the differences in the application of tox endpoints from the other ICH guidances listed here. The USP monographs that are listed here are also quite extensive. Some of these are not even promulgated, As you can see with the 661.2 and 665, those are still off in the distance. But in our experience, expectations towards those ends are still being discussed through FDA communications, whether that's IRs or CRLs. And many sponsors are starting to move to execute those, even though they are draft documents, for the quality control of their product. Some other guidances in the space for single use, the BPOG protocol here in the top left, originating in 2014, and some evolution and reiteration of those documents since then. At the bottom left, you see 665, again in draft, but certainly being applied in the space, even in draft format with risks, strategies, and some analytical techniques built in. And then BPSA on the right here for extractable leachable considerations for cell and gene therapy in the space. To add to the technical complexity here, on the left we present a list of common polymers, and other than a lengthy list here, starting with poly, certainly these materials are radically different. They're used for different end uses, and their chemical composition and even their load of additives is quite different. So there's a different risk associated with each of these polymers. And again, experts in the field would recognize those risks and design experiments to try to address risk-based questions and help to mitigate those risks where possible. The ultimate question is why? Why do we find ourselves in this conundrum? And really, it starts with the idea that polymers themselves as pure materials are unstable. Additives are intentionally added to improve the quality of those materials. And so those additives or stabilizers are low molecular weight compounds relative to the polymer itself. As a result, they have the capacity of migrating out of the plastic and potentially into the drug product or the manufacturing systems process stream. And it's understanding that process and characterizing the outcome of that process that really creates an extractable package that can better inform what leachable testing needs to be performed. Pharmaceutical formulations, for example, can be good solvents for plastic additives, perhaps high in organic content or having other composition that would facilitate extractable migration out of that plastic component. And the question that we get asked a lot, what components should be evaluated? Well, certainly anything that's in contact with the drug product itself should be part of that investigation. This is where the extractables become leachables, is at the surface of that direct contact. Any components in the processing stream that come into direct contact, anything in the drug production and filling lines are also prone to the introduction of compounds that, quite frankly, don't belong in the final drug product and can be either toxic or indirectly toxic adulterants. An example here of a risk table, this is taken from the BPSA guidance, and somewhat simplistic. The first question to answer relevant risk is, do you have extractable data? Certainly if the answer is no, feel free to reach out to us and we can help provide that with a detail-oriented extractable study design. But ultimately, you need the extractable package, that library of compounds, in order to assess potential risk to an end patient. So ultimately, when that is generated, the question is, in a worst case scenario, how that extractable study should be designed, can you assert patient safety based on that information? There are some circumstances where sponsors are able to submit those findings directly to the regulatory bodies and make a justification case that all safety endpoints are covered and that leachable work would not be necessary to be performed. More often than not, there are some problem actors, chemicals that may present a safety risk, and a more defined quantitative determination of how much of that extractable compound becomes a leachable compound and what the true risk to the patient would be. That is the tendency, and we see sponsors typically moving into that determination to develop and validate leachable methods and have quantitative certainty around those endpoints. So a little bit of summary here. The extractable study is what could come out. But these bullet points, you'll see these concepts in the guidance, such as USP 1663, are referring to independent study designs that are asking different questions. But no matter what question is asked, the extractable library that's generated is what could come out under that set of conditions. The leachable study, we would be testing the drug product for any of these compounds or even unspecified compounds that could present themselves as adulterants to that final drug product. And so, toxicity, as we've talked about, but also the potential of chemical interaction. There are plenty of case studies by which the active in a drug product is actually part of a chemical reaction and renders it unusable for the patient's application. Physical interaction can be as deleterious. Certainly, a transfer of material components into that pharmaceutical product, whether toxic or not, is a quality concern and something that could be addressed in an iterative capacity. Finally, here, and something that is commonly either ill performed or not performed at all by the sponsors of record, is the idea of correlation. It is an expectation in the regulatory guidance to know where these compounds came from, to correlate an observed leachable back to its extractable origin. And this not only allows for a demonstration that methods are correctly in place for the purposes of quality control, but also to inform the sponsor if a change needs to be made, that we know exactly what component these compounds came from and can make an educated decision towards that end. It allows also for the mimic of leaching so that quality control may be performed at the extractable level. And ultimately, this is above and beyond the toxicity concern. This is a quality concern. An overview here of the extractable assessment. For the overview, I won't read all these in detail, but I think, again, it speaks to the idea that there are a lot of checkpoints and decision points in the establishment of a comprehensive extractable study. So working with the experts in the field and addressing relevant and constantly changing regulatory expectations is a pathway to success. And that risk evaluation then becomes a leachable question of concern. If the program moves in that direction, understanding interaction with the drug product, understanding and designing appropriate duration of contact. In some circumstances, you may design a study that invokes Arrhenius extrapolation so that information can be gleaned in an accelerated study to compare against or to supplement real-time establishment. As mentioned, in engagement with the toxicologist to establish safety endpoints and margins of safety. And a lot of this has to do with the route of administration. As we saw in that complex table a couple of slides back, understanding the route of administration can also funnel into some of the relevant regulatory guidance documents that are out there and design of experiments. As I summarize a little bit here before I turn it over to Dr. McGee, just a little bit of compare and contrast for the purposes of EL efforts for container closure systems and how that may be similar or different to those for the single-use space. For container closures here, extractable studies are a necessity. Under exaggerated but relevant conditions, studies can be designed with solvents of higher solvent strength than the formulation and a slightly higher temperature if necessary. Analytical methods invoked use GC and LC chromatography with different sample introduction for organic, extractable, and leachable compounds, as well as elements by ICP-MS. These studies should be designed around a relevant AET, that threshold for reporting and structural determination for the purpose of safety by toxicology. And those extractable efforts will fit into a leachable study to inform target compounds. But again, leachable studies need to be designed around the purpose of also screening for unspecified compounds. When you end up with one of those potential compounds that is either not structurally defined or does not have correlation back to the extractable library, further efforts are necessary. but similar techniques, similar methods. In the single-use space, we would be typically performing simulated in-use leachable studies to help inform potential leachables in an extractable determination. And similar analytical techniques would be invoked, similar endpoints. The AET is, of course, relevant to the patient from a leachable exposure standpoint. So we'd still need to design experiments with that determination in mind. I will turn it over here to Dr. McGee.
Thanks, Chris. What we've seen so far has been a very classical interpretation of a series of definitions and the current state of the industry, particularly from a container closure standpoint. But there's something I want you to keep in mind, and those 4 words I think I can't overestimate, and that is suitable for intended use, which essentially comes up with the concept of risk. Most of extractable leachables is really about risk. And you can ask yourself, how did we get here? If you go back to classical manufacturing of small molecule pharmaceuticals, and you took a look at your manufacturing facility, chances are you'd see rows and rows of stainless steel or glass vessels, which were generally considered inert when it came to organic contaminants. Classical biopharma really changed all that as they started to increase the adoption of single-use systems or single-use technologies, which really made the manufacturing process more flexible as they were based upon disposable components. And that's where the polymeric piece really began to explode. Standard protocols have been published. A lot of work has been done on the subject. But there's something to keep in mind for suitable for its intended use. When you were working in classical biopharmaceutical manufacturing, right, the cells were utilized for the production of the drug substance. If you were looking to produce a protein or an antibody or an enzyme, chances are you had other production steps which would allow you to filter out any potential contaminants. You could perform ultrafiltration, you could perform sterilizing filtration, you could perform centrifugation, and a number of other steps to potentially eliminate the possibility of contamination from the manufacturing process entering into your drug substance. Things become much more complicated when you start to look at cell and gene therapy. Here in cell and gene therapy, generally the cells are the drug product, and there really is not much opportunity to separate impurities from the product. So back to suitable for intended use. You have to take a look at your components in the manufacturing process, right? Because they are all exposed to the process stream, and that process stream is what's going to extract any potential impurities from the components. If those impurities survive the entire process and make it to the final step, that gives them the opportunity to accumulate in the drug product. So pearls that can persist to the drug product are then essentially leachables. So all of this contact between the process stream and the components has to be evaluated, and that determines your risk. From a regulatory perspective, the agency is going to review your submission, and they're going to be looking at compatibility of your components with your process. A lot of manufacturing processes, particularly in biopharma and cell and gene therapy, are considered complex. There's a lot of information to be gathered. Duration of the process, holding times, flush volumes, all of this goes back to compatibility and suitable for intended use. A lot of conversations need to be had between the chemist, the engineer, the biologist, the toxicologist. Everyone with a stake in the process needs to provide input. You have to come up with a detailed qualification procedure and produce the data to justify the equipment, the components, your manufacturing process. Again, it goes back to risk. Risk can be anywhere from worst case to actual case, but it can never be zero. It can be reduced to an acceptable level, but everybody's version of acceptable is different. Each situation can vary. In some cases, your risk might be deemed unacceptable. So how do you perform a risk assessment? You can start by browsing through regulatory guidance, national and international standards. There's plenty of compendial monographs, best practices, and other publications available for you to consult. BPSA is one such industry group dedicated to expanding bioprocess adaptation and use of single-use systems. Here's another example of a decision tree to perform a risk assessment. The guidance from BPSA basically began years and years ago for classical biopharma and the use of single-use systems. And essentially what you'll see is identifying your material of composition. Is that material compatible with your process stream? What are the temperature and time that your process stream is in contact with that material? What is the surface area? Are your components pre-treated in any way? All of this information needs to be added to generate your risk assessment. Chris presented earlier a list of polymers that are typically found in the industry. Here's a list of common equipment used in biopharmaceutical and cell and gene therapy, ranging from storage containers and bags and reaction materials to pump services, tubing, gaskets, O-rings, filtration columns. All of this equipment, be it polymeric based, has the potential to release common extractables into the process stream. We know a lot about common extractables from previous work with container closure systems in particular. Antioxidants and their degradants, plasticizers, monomers, processing aids, lubricants, UV absorbers, all of these things can potentially leach into the process stream. You have to keep in mind the proximity of the component to the final product. You have to evaluate the ability of the process stream to extract. That involves contact time. That involves contact temperature. That involves surface area. Again, pre-treatment. All of this goes to material compatibility. All of this is potentially a large part of your risk assessment. BPSA brings up a lot of good points in their paperwork on biopharma manufacturing and cell and gene therapy in particular. You know, the idea that certain leachables can have safety implications to our patients, particularly patients that are already compromised, be it a lack of organ function or a compromised immune system. Compatibility issues can be introduced by the presence of leachables via chemical reactivity or potentially cell toxicity. You know, is it possible that your leachables are actually initiating a cancerous change in your cells through some sort of genotoxic or mutagenic effect? You know, the risk has to be evaluated, and that starts with a classical extraction study. Some of the materials that you'll commonly find in cell and gene therapy, particularly bags and tubing sets and expansion containers and media containers that are used, are they suitable for their intended purpose? This material includes PVC and ABS and EVA and PE and PETG and all of those polymers that we've seen that have the tendency to be extracted by your process stream and generate leachables. So where are we at? Given the extensive use of single-use systems throughout cell and gene therapy manufacturing and classical biopharma in general, you know, the cell-based products and the variety of possible contact surfaces and contact time, you can rest assured that they will be impacted by leachable compounds. Because manufacturing is highly dependent upon these polymeric materials, risk assessment needs to be performed on the process stream and its ability to leach materials from these components. And also keep in mind the difference between classical biopharma production and cell and gene therapy is that in this case, the cells are the product. And apart from a few washing steps, there's really not a lot of opportunity to separate any potential impurities. Regulatory guidance. Everyone turns to regulatory guidance to support their risk and justification statements. In general, some may say or be of the opinion that the regulatory guidance is absent. So we draw upon previous guidance on container closure systems and initial use of single-use systems. Of particular importance as far as regulatory guidance goes are the USP chapters 665 and 1665. They really are the benchmark supported by the regulatory industry for an extractable investigation. Prior to the publication of USP's monographs, the Biopharm group under the BPOG protocol, what has become known. Chris mentioned it was originally published in 2014 and then updated again in 2020. Biopharm set out to standardize extractables testing for single-use systems. They gave essentially a blueprint, a set of directions for you to generate and report data on components. And this data then being standardized could be comparable. The protocol was good for all dosage forms and all routes of administration. Again, it was about understanding your risk, assessing your risk, understanding your process, your formulation, the ability of your process stream to extract. It's leachable power, really. You know, a lot of manufacturers nowadays are able to provide you with some information on extractables. The vendor can provide this compatibility information for your review. But you have to remember that every process is different. Every product is independent. And there's a very good possibility that the vendor provided information is not suitable for your intended use. You need to justify that by producing your own data. The BPOG protocol gives you some instructions and recommendations on how to perform such an extractable study, including temperatures, extraction solvent, surface area to volume ratios. Variability in lots and resins and things of that nature can be addressed. Time for extraction, incubation, as well as the solvents to use. The latest version of the BPOG protocol gives you 4 solvents to consider, including 50% ethanol, water, an acid solution, and a base solution. All of this is necessary to generate that library of extractables that could potentially become leachables. 665 as the published monograph draft kind of builds along that idea by generating 3 different solutions. Again, a 50% ethanol solution, an acidic solution, and a basic solution. And they also incorporate the concept of risk, which has become so important in the industry. Judging that risk from low, medium, and high allows you to tailor your studies to what solvents you're going to use and what testing you're going to perform. Some of the highlights from 665 are its standard extractables protocol. Providing the ability to simply use 50% ethanol as an extraction solvent in lower risk situations and do a subset of testing that's generally considered more qualitative than quantitative. For moderate risk situations, you start to get into your controlled extraction study, where you're actually looking to generate your extractables profile, but this case using 50% ethanol as your only solvent. In your higher risk situations, you add an acid solution and a base solution to try and bracket your process stream. So USP has taken the approach pretty much down the middle rather than worry about the worst case. Obviously, if your process stream is outside of the middle, if you are running something that's more acidic than pH 3 or you are introducing something more basic than pH 10, you have the flexibility to adapt your extraction solvents to something more suitable to your intended process. Again, we have to go back to suitable for intended process, right? The chemical and physical nature of the component and its propensity to be leached, the chemical and physical nature of the process stream and its ability to extract, the conditions for manufacturing that are the driving force for generating leachables. And you can't fully characterize the situation without considering the ability to actually provide clearance. Are your components further upstream so that they have the chance to either be removed or potentially diluted so that an adverse effect is unlikely when you get to the final step? You must consider all of these possibilities in your risk assessment. And one further thing is the clinical use of your product, right? If your product is intended for a one-time use and it's just a small volume of material, your risk is certainly different from someone who is clinically using large volumes of material over many days up to a lifetime. All of these conditions and considerations can be used to produce a risk assessment. USP 665 gives you the opportunity to perform a risk evaluation matrix, where they primarily look at 4 different categories: the duration of contact between the component and the process stream, the temperature at which that contact occurs, the chemical composition of the process stream, and of course, the component's material of construction. Obviously, some of this is pretty intuitive, right? The longer your process stream is in contact with the component, the more a chance an extractable can be released from the material and become a leachable. The temperature contact is also important, right? The motion, kinetic motion in material is higher at higher temperature. And chemical composition. The presence of organic solvents leads to a higher ability to extract from the material. So is your process stream aqueous, or somewhat organic, or highly organic? Do you use surfactants in your process stream? Do you use blood or blood-derived substances in your process stream? What about lipids and proteins? All of that needs to be considered to determine your risk. From the standpoint of material reactivity, you also have to take a look at the total level of additives that are available in your component. Has your component been irradiated? Is it subject to gamma irradiation or X-ray sterilization, or do you autoclave with steam? Has your material been chemically treated prior to use? All of these considerations need to be evaluated. Do you use glue, bonding solvents, adhesives, any chemical means of connecting component materials together to produce assemblies or sub-assemblies? Again, all of this becomes part of your risk assessment, intended to prove suitable for intended use. You can combine all these factors and come up with a numerical score to determine whether or not you're in a low-risk category or a high-risk category or somewhere in the middle. But at the end of the day, you still need to generate the data. You still need to understand your process. USP 665 falls back on container closure guidance, particularly USP 1663, for generating and analyzing the extracts. The process of assessing extractables begins with scouting. It moves on to discovery and identification. Hopefully, we'll cross our fingers at that point. Quantitation, as well as assessment. So for assessment, your extractable profiles have to be interpreted, right? Because remember, this is all about patient safety. You're establishing risk. Most likely, a chemist is not equipped to establish risk. You need to bring in toxicological experts to evaluate and assess the data. The assessment has to consider the safety risk of each individual relevant extractable as if it were a worst case leachable. And keep in mind that it's not the vendor's responsibility to perform the safety assessment. It really is the applicant. He or she or they who are securing and owning the regulatory approval of this drug product is responsible for the safety assessment. And the safety assessment has to be backed up by data. Here's a selection of important references that can be easily pulled from the web. And what you'll see among the titles is risk. So you'll see a lot of work being done about the identification of leachable compounds that can be detrimental to cell growth. How many leachable compounds can be found that can actually prevent cell growth? When you're working in biopharmaceuticals and your first step or your initial steps have to do with cell growth, and now you have a leachable that's hindering that cell growth, more and more importance has to be put on to the characterization of the materials of construction being used for the components, and that there is suitable for intended use. Latest thoughts. BPSA has done a great job in the cell and gene therapy space for providing thoughts for everyone to consider. Current research trends. Again, a review of current literature would tell you that pearls may be detrimental to cell growth, including expansion and viability. There's also some belief that they may cause undesired effects of stimulation. All of the extractable testing we've talked about to date still doesn't address another potential question. If your extractables remain in your product and become leachables, could they be absorbed or adsorbed by the cells and then influence product quality and patient safety? Could they manipulate the cells to produce degenerative cells? These are things that have to be considered, but are not necessarily answered by extractable studies. Some of the things in the literature that have identified possible bad actors really go back to the idea of the degradation of antioxidants. A lot of polymer material can use a phosphite-based antioxidant. The degradants tend to be phosphate-based, phosphite to phosphate. Some of these have been demonstrated to inhibit the growth of cells or perform other undesired effects. We also have a case in the literature of 3,5-dinitrobisphenol A, which is interesting that a BPA derivative potentially found in components used for mixing media could contribute to a decrease in cell growth. And then PVC itself is another polymer that's kind of in a special case because that material can contain upwards of 30 to 40% phthalates. And phthalates have been shown to have an effect on the growth of cells in media. So in summary, I think it's necessary for everyone in the biopharma space to perform an E&L assessment of their process. Not only do I think so, the regulatory authorities think so. You have to guarantee that patient safety, always, patient safety comes first. So to perform that E&L assessment, you can consult guidelines, you can map out your process, you can perform your risk evaluation, you can execute those laboratory studies, you can consult with your toxicologist, and most importantly, you're going to prepare all of your proper documentation and justify everything that you've done so that you can convince the regulatory authorities that your product is safe and that the materials and components that you used in your process are suitable for their intended use. E&L studies are a regulatory requirement for drug product submission and approval. That's not debatable. All polymers and plastics can be a source of extractables and potentially become leachables, adulterating your product. That's not debatable either. E&L studies should be designed in line with promulgated parameters, and you have to consider the risks. Your study design has to look at potentially absorption and direct transfer. And most importantly, I think what we've demonstrated today is that E&L study design is increasingly complex. It can be very time intensive, and it's going to require a highly skilled and technical team to conduct these investigations and prepare the data for assessment. Therefore, you need to involve the experts. And naturally, that's where we come in. Here at Sylvius USA, we have a track record and a long-standing reputation as a center of excellence for extractable and leachable studies. We're located just southwest of Boston at our Canton, Massachusetts facility in the USA, performing extractable leachable studies for over 20 years. We have a long suite of equipment available, including high-resolution accurate mass as necessary. But most importantly, we're part of a larger group, the Sylvius Group, founded over 20 years ago in Switzerland. That's our main office and the location of a majority of our scientific staff. So not only do we have scientists here in the United States, we have scientists across the world in Switzerland and the Netherlands and France, ready to support your next study. Our track record and expertise, again, goes back over 20 years in a variety of classical and future lost my train of thought. Synthesis, ligands, small molecule analysis, analytical chemistry, medical device testing, inhalation products, biopharmaceuticals. Basically, we are looking to be your one-stop shop for help. Just about anything that you can come up with in the analytical chemistry space. I thank you for your time and attention today, and Chris and I are happy to take any and all questions.
And thank you very much for this presentation. Just a reminder for the audience, to ask questions, you can send them via the questions widget. Just type them in this box and click submit. Please note that you can still send your questions after the webinar and they will be answered at a later date. And it looks like we've got several questions already. And our first question is, how long is needed to complete EL testing?
Thanks, Eugenia, and thank you to whoever prompted that question. Honestly, it's a question that we get a lot, and to not hedge bets too much here, I think it depends on who you ask. We have colleagues in the space that have responded to that question with "it takes as long as it takes," and I think we'll leave it as it depends. It really depends on what the end points of those determinations are. From start to finish, EL is a series of studies. It's not a checkbox activity. And so to recognize it as such, to generate an extractable profile that's meaningful to risk assessment and potential information for a leachable study, you could be looking at on the order of roughly 6 months from the start of extractable activities to actually having validated leachable methods for quantitative accuracy and precision of control of target leachable compounds. And then, of course, the execution of a long-term leachable study to answer questions about shelf life or even in use, may have different time domains. We recognize that sponsors don't like to hear uncertainty in a direct question like this, but I think what we've demonstrated today that EL is really, it's an iterative process. It's a life cycle process. And ultimately, any attempts to cut down on that analysis, taking shortcuts or rushing through, is usually a recipe for a CRL, and certainly recognizing that could also lead to significant loss of market share and revenue. So again, I think the answer to the question is it depends. But certainly it goes back to the idea of engaging with experts and designing something that does answer that question of suitability for use and patient safety.
Thank you very much. And we have another question from the audience. Do I need to use multiple analytical techniques, or can I pick one?
That's another good question. I think it's if we look back to one of the slides that I had presented, there are 3 typical chromatographic techniques that govern the determination for organic, extractable, and leachable compounds. And each of them plays a different role in casting as wide a net as possible. That typical gamut includes LCMS for non-volatiles, direct inject GCMS for semi-volatiles, and headspace GCMS for volatile compounds, whether for extractable or leachable determinations. And all of those in conjunction should allow for the greatest opportunity for discovery of compounds. And then, of course, from a leachable standpoint, it is still a regulatory expectation that all of those methodologies be invoked so as to potentially discover unspecified leachables and react to those during a typical leachable study. So the short answer, I would think, is no. And unfortunately, all of those methods are necessary for a comprehensive covering of organic extractable leachable compounds. And then certainly for inorganic extractables or leachables, these are elemental. That needs to be performed by a separate technique, typically is ICP-MS.
Thank you very much. And our next question is, which is the best approach in the frame of the leachable study?
It's quite a loaded question. I think ultimately it depends also on the nature of the product. One of the things I think we've demonstrated here is that a comprehensive understanding of the manufacturing process and that process stream and potential inclusion, it would be a strong recommendation of ours to execute those activities prior to a leachable study, so that direct information can be provided to the analytical chemist as they embark on leachable method development and validation, so as to capture with quantitative certainty an analytical output for those potential pearls. The leachable study in and of itself is strongly dependent on multiple factors, but the shelf life of that end product, the nature of that product. There are circumstances by which the guidance actually allows for a modification of a pivotal leachable study based on whether or not that end product can be used in an analytically expedient fashion. So again, ultimately here, I think it's a point to engage with the experts for the proper design of a leachable study. But again, not taking shortcuts. Depending on the nature of that product, it requires careful justification to design a leachable study in such a fashion that does not meet typical regulatory expectation.
Thank you very much for the response. And we have another question from the audience. Right. You mentioned that the pearl isn't expected to grow as a function of leachable stability time points. Can I use that to categorize a compound and avoid further extractable testing?
So that's actually a really good question. I think what the question is indicating is, if you run a leachable stability study and you see that there's a compound that has a static concentration as a function of time, can you automatically put that into the category of a pearl and just say it must be coming from my manufacturing line, and therefore I have some measure of control over it, or at least an understanding of where it may come from? We haven't seen any success with that type of approach in our experience. So I'd say it's typically not enough to categorize the compound. I think that you could probably construe other circumstances that may generate, let's say, a peak of interest here in a chromatographic profile that doesn't change significantly as a function of time. Beyond that, I would remind that one of the goals here of the cyclic EL process is really that of correlation. By saying that, well, we know it's coming from the manufacturing line, that's really insufficient based on the guidance and expectations that have been presented today. I think it is critical to be able to tie back to an individual component, and ultimately that burden of proof is on the sponsor of record to execute with certainty that they have control and could modify that if mitigating factors are needed.
Thank you very much. Okay, we have one more question from the audience. Right. Can I run my extraction study with Can I run my extraction study with finished drug product and use this to establish patient safety?
Also a question that we get a lot, and this is, I think, an example of trying to simplify and expedite the process by jumping to the final conclusion. Ultimately, the answer is maybe. This can be a strategy as a supplemental study to help, I think, to better inform and may be used as a risk mitigating factor to perhaps better understand the potential leachable profile by using the formulation as the extracting solvent. But I think that we've clearly laid out today that it does not fit the regulatory expectation. Casting that wide net in the establishment of the extractable library is really step one, no matter what, and that is expected to be performed at the very least supplemented by analytically expedient or facile solvent systems. as you can imagine, most of the drug product matrices that you could work with have complexity. In some cases, are incredibly complex in nature. Pure organic solvents that would typically be used in a controlled extraction study do not have that complexity, and as a result are going to yield the greatest amount of information when those extracts are evaluated on a chromatographic system. Another way of saying that is that your drug product could actually cover up some of the potential compounds that we would be interested in to track as potential leachables downfield. The ability to define what can come out is not something that your drug product would typically allow in an analytical capacity. And again, ultimately, it doesn't cover the expectations of the regulatory bodies. The idea of using it as a simulant is certainly something that's covered in the regulatory guidance, but also requires careful justification and can't stand alone as a study. And finally, the expectation really is to go and validate these methods for some subset of target leachable compounds. And that would be the step in which the bulk drug product would be used to demonstrate the suitability of the analytical methods for their intended use. And that is the quantitative determination for the purposes of informing toxicology and informing further quality control mitigation.
Thank you very much. And our next question is, how do you address leachables that don't have a direct correlation to the extractable library?
Well, certainly this can happen. I think we showed on one of the early slides that partial lack of overlap between the extractable library and the leachable library. And I like the fact that we're using the term correlation here. That correlation piece is critical to the iterative understanding and control of patient safety through leachable to extractable correlation. So that process is not always straightforward. It's possible, let's say, that an additive has degraded in the presence of the drug product and has become a unique chemical entity. It's possible that that unique chemical entity is not seen in its in that particular form in the extractable library. But certainly working with experts in the fields and those that understand organic and analytical chemistry, it is possible to be able to tie that correlation back to the parent extractable compound. So having a fundamental understanding of the chemicals that we engage on a daily basis, and even some that are novel to the situation. I think the other difficulty is the subset of compounds that can react with the drug product, and those would be very unique. They'd be unique to a specific sponsor of record's formulation, and have to be investigated, using more sophisticated techniques. It becomes an investigation, and again is an iterative activity to better understand and better control the safety endpoints for patient safety. Looks like we've got time for one more question.
Oh yes, we have one more question from the audience. Is the LCMS used in your EL screening study a TRAP or a TOF?
Excellent. Well, we have capacity for both MS techniques here on site. And I think really the question kind of speaks to the higher order concept of high resolution accurate mass spectral determination. It is critical in these studies to understand the structure and have confidence in the structure of an extractable or leachable compound so as to better inform toxicology. USP 1663 speaks to those confidence thresholds for identification. And certainly the earlier that you identify a compound, the better it can be controlled. So the answer is yes, high resolution accurate mass techniques are a must, and we employ them on a regular basis for extractable and leachable studies.
Thank you very much, and just a reminder that you can still send any questions while watching on demand. All questions will be answered at a later date. I would like to thank Christopher McKee and Christopher Letandres for this presentation and Sylvius for sponsoring this session. To the attendees, you will receive an email telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is pmi-live.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned. Thank you once again, and I hope you all have a nice day.